In 2015, scientists from Simon Fraser University did something most people would consider deeply unsettling. They took a dead pig, weighted it down, and dropped it 300 meters into the ocean off British Columbia. Then they pointed a camera at it and waited. What happened next was so fast and so brutal that it forced them to completely redesign the entire experiment. This wasn't some morbid stunt, it was real forensic science, using pig carcasses as stand-ins for human bodies to answer a question police in this exact region desperately needed answered. Welcome back to the Abyss Report. This is the strange, real story of what actually happens when something sinks to the bottom of the ocean. The research was led by forensic scientist Dr. Gail Anderson at Simon Fraser University's Center for Forensic Research. Her question was simple, but forensically critical. What actually happens to a human body once it sinks into deep, cold ocean water? Until this study, almost nothing about marine decomposition was understood in any real detail. Most of what investigators knew about how bodies decay came entirely from research done on land. But when police recover a body from the ocean, that land-based knowledge simply doesn't apply. So Anderson's team partnered with Ocean Networks Canada, which operates an underwater observatory called Venus in the Strait of Georgia. Venus gave them something no forensic study had ever had before, a live, cabled camera and sensor network sitting on the ocean floor itself. They could measure oxygen, temperature, salinity, and pressure continuously, while filming everything in real time. Pig carcasses were chosen because they're widely accepted in forensic science as the closest available proxy for human tissue, skin, and internal bacteria. The team lowered a carcass down to 300 meters and settled in to observe. They expected a slow, gradual process, similar to what happens on land. What they got instead was almost nothing like that at all. In an early trial run, before the main study even began, researchers lowered an exposed carcass with no protection whatsoever. Within hours, blunt-nosed six-gill sharks arrived. These are large, ancient-looking predators that can grow over 15 feet long, common in the deep waters of the Strait of Georgia. The sharks tore into the carcass aggressively, consuming large sections of it almost immediately. For a controlled scientific study, this was a disaster. If sharks could destroy a carcass in hours, there was no way to observe the slower, more detailed stages of decomposition researchers actually needed to study. So the team went back to the drawing board. They designed a special platform holding two matched carcasses. One left fully exposed, the other protected inside a barred metal cage. The cage blocked sharks completely, while still allowing smaller scavengers like shrimp and amphipods to move freely in and out. It was essentially an underwater science experiment, engineered specifically to outsmart a shark. With the sharks controlled for, the real discovery came almost immediately. Not from a predator, but from something far smaller. Within minutes of each carcass hitting the seafloor, swarms of tiny shrimp-like crustaceans called lysianacid amphipods arrived by the thousands. They poured into every opening on the carcass, the mouth, the eyes, any wound or natural cavity, and began feeding from the inside out. Unlike the sharks, the amphipods were relentless, methodical, and impossible to stop. In the fall trial, they stripped an entire carcass down to bone in just 3 days. In the spring trial, it took 4. That's a full pig carcass, reduced to a skeleton, faster than most people finish a long weekend. Researchers also recorded something unexpected happening in the water itself during peak feeding, a sudden, sharp, localized drop in dissolved oxygen levels. It coincided exactly with the amphipod swarm, likely caused by the sheer biological activity of thousands of tiny bodies feeding at once. The cage that kept sharks out did nothing to slow the amphipods down at all. If anything, without shark interference, the amphipods worked even faster and more efficiently. The ocean's smallest scavengers turned out to be far more dangerous to the experiment than its biggest predator. Because the Venus Observatory recorded continuously, researchers had something forensic science had never had before: a minute-by-minute visual timeline of decomposition happening in real time. They watched skin discolor and split within the first day. They watched internal tissue disappear from the inside out, hollowing the carcass while the outer shape briefly remained intact. They watched the carcass shift and roll slightly across the seafloor as scavengers worked beneath it. Every stage was time-stamped, measured, and cross-referenced against oxygen and temperature data collected every single minute. This level of continuous, high-resolution data simply didn't exist for marine decomposition before this study. For investigators, that data is invaluable. It means that when a body is recovered from cold ocean water, forensic teams now have an actual scientific baseline to estimate how long it's been down there. Before this research, that same question would have been little more than an educated guess. Now it's backed by real, time-stamped underwater evidence.
Here's what made the Strait of Georgia results so surprising. Earlier studies by the same research team, conducted in nearby Saanich Inlet at around 100 meters, had shown something very different. In that lower oxygen environment, carcasses could survive largely intact for weeks, sometimes months. But the Strait of Georgia site was deeper, more exposed to open currents, and far more oxygen-rich. In that highly oxygenated water, the same size of carcass could be completely skeletonized in under 4 days. More oxygen meant more active scavengers, and more active scavengers meant a dramatically faster disappearance. This told researchers something crucial. There is no single timeline for underwater decomposition. Depth, oxygen levels, currents, and even the season can each independently speed up or slow down the entire process by weeks. A body in one part of the same coastline could vanish in days, while an identical body just a few miles away could remain recognizable for months. For forensic investigators, that variability is exactly why this research mattered so much.
And this is where the story connects to something you may have actually heard about. Since 2007, more than 20 detached human feet still inside sneakers have washed ashore along the coastlines of British Columbia and Washington state. For years, the discoveries fueled wild theories, plane crashes, serial killers, even conspiracy theories involving foul play on a massive scale. Local investigators needed real answers, and much of that answer came directly from the same decomposition research. Dr. Anderson's work showed that the ankle is a relatively weak point in a decomposing body. As tissue breaks down underwater, joints loosen, and the foot can separate from the rest of the leg entirely on its own. No cutting, no violence required. Here's the part that explains everything. Modern running shoes are built with buoyant foam. Once a foot separates inside a shoe, that buoyant foam keeps it afloat, while the rest of the body, without that flotation, sinks or drifts elsewhere entirely. The shoe also protects the foot from further decomposition and from most scavengers, which is why they're often found remarkably intact. Currents throughout the Salish Sea then carry these floating shoes toward shore over time, sometimes taking months, sometimes taking years. Investigators eventually used DNA testing to match many of these feet to specific missing persons. In every single case examined by the BC Coroner's Service, foul play was ruled out entirely. Most were links to accidental drownings or previously unreported missing persons cases. A phenomenon that once fueled genuine public panic turned out to have a scientific explanation built directly on pig carcass research from the ocean floor. Put it all together, and the picture becomes clear. A body enters the water and begins to decompose exactly the way the Venus Observatory research documented. Depending on depth, oxygen levels, and season, that process can take anywhere from days to months. As decomposition progresses, weaker joints, especially the ankle, separate first. If that foot happens to be inside a modern, buoyant running shoe, it floats, while the rest of the body does not. Ocean currents specific to this coastline consistently carry floating debris towards certain beaches again and again. That's not a coincidence, and it's not a serial killer. It's oceanography, forensic biology, and a very specific kind of modern footwear, all combining in one very unusual pattern. This case is a genuine example of real forensic science solving a mystery that once seemed impossible to explain. And it started with something as simple and as strange as lowering a dead pig into the ocean and watching what happened. Every uncomfortable detail from that experiment ended up mattering months and years later in real investigations involving real people. That's the part of ocean science nobody expects going in.
A dead pig dropped 300 meters into the ocean, testing sharks that gave up early and amphipods that didn't. A skeleton in 3 days. An oxygen crash no one expected. A forensic mystery quietly solved years later using exactly this kind of data. The deep ocean doesn't just hide strange creatures. It runs its own rules on time, decay, and evidence. Rules police and scientists are still only beginning to understand. If a pig carcass experiment could help solve one of the strangest coastal mysteries in North America, it makes you wonder what else the deep ocean is quietly explaining if anyone bothers to look. This topic touches on real missing persons cases and real loss. We've tried to handle it with the respect it deserves. Subscribe to the Abyss Report. Next week, we go back down. See you in the deep.